So we were talking about transcription elongation in eukaryotes, and that's where we need to pick up. So let's talk about the RNA polymerase II again, right? You remember RNA polymerase II, the one for mRNA, messenger RNA, right? Okay, now RNA polymerase II, it does not proceed just like nice smooth cruising along the DNA. Instead, it's really jerky, right? Back, forth, back, forth, back, forth, back, forth, right? Do you remember why? That's right. It's that proofreading in the 3' to 5' direction, right? And, remember, RNA polymerase II is associated with all those general transcription factors during initiation. Those were called initiation factors. We can call them that, right? Well, elongation of transcription also has general transcription factors, and we call them elongation factors. Okay, so the RNA polymerase II, it's not alone. It's got a whole pile of proteins stuck to it, helping it to cruise through the DNA. Okay, now RNA polymerase II has a problem. The problem is basically it's like, it's trying to plow its way through this DNA, but because the DNA is a helix, all this tension builds up in front of it, right? And it induces these positive supercoils ahead of where the transcription is taking place. And then, because it's moving forward in the 5' to 3' direction, we get all these negative supercoils behind it. Okay, well, luckily, there's a fix. And in eukaryotes, they use the topoisomerase enzymes, which I'm sure you remember from DNA replication. That's right. The topoisomerases, they relieve the tension ahead of the transcription site. And in prokaryotes, they have a special topoisomerase. It's called DNA gyrase. That's right. It does a little gyration, right? Little gyrase unwinds the helix ahead of it so that transcription can take place. Okay? Now, the thing about eukaryotes is, like I say, we're complicated. We don't just transcribe the DNA and make our little RNA transcript and leave it at that. No. The RNA transcript we made, it's a preemie, right? It's just a little baby preemie. That's right. It's a pre-mRNA. So now what we have to do is we've got to process this pre-mRNA so it can mature, so it can grow up, right? And be all that an mRNA transcript can be. Okay? So in transcription in eukaryotes, the transcription elongation is actually tightly coupled to RNA processing. Okay? And what I mean by processing is that the mRNA gets modified. Okay? And it gets modified in really 3 main ways. One, it gets a cap on the 5' end, right? A cap, kind of like a little hat, okay? And then it gets a polyadenylation or a poly-A tail, a polyadenylation on the 3' end of the mRNA transcript, right? A little poly-A tail. So it gets a cap on the 5' end and a tail on the 3' end, right? Sort of like a little baby tadpole, okay? Now, and the reason you need these 2 things on the mRNA is because you need to determine, if both of the ends are present so that there's sort of like, a top and a bottom or, a forward end and a hind end, because these mRNAs, where are they being made? Where does transcription take place? Let's do a little cellular geography. Where does transcription take place? That's right. Right inside there, right? It's taking place in the nucleus, where the DNA is, right? In the nucleoplasm. Okay? So these pre-mRNAs, when they're made in the nucleoplasm, we got to get them out of the nucleus into the big bad world of the cell, which means they have to cross that nuclear envelope, right? Can you imagine how scary it must be to go from the nucleoplasm into the cytoplasm? Wow. Anyway, to get there, they have to go through a little portal, right? A little portal, that nuclear pore that we talked about, to suck those pre-mRNAs right out of there, right? But before they go out, they better be mature enough to survive, which means that they have to be pulled out. And those nuclear pores, they're not stupid. They're going to look at the pre-mRNA and they're going to see, are you grown up enough to survive in the big bad world of the cytoplasm? And the way that they know that is they look for the polyadenylation tail. And they're going to suck them out. Tail first, right? Okay, so that's the second thing. 1, they get modified with a cap on the 5' end. 2, polyadenylation of the 3' end. And 3, okay, the mRNA actually has 2 different types of coding regions in it. They're called exons and introns, okay? The exons are the actual regions of the mRNA that directly correspond to the amino acid sequence of the protein that they code for, that they're a script for making, okay? Those are the exons. The introns, those are the portions that actually end up getting cut out of the pre-mRNA, okay? So what happens is these exons are sort of situated within the mRNA, and between them are introns, and the introns get cut out, and then the exons get squeezed together, okay? Now, in order to do this, we call this RNA splicing. And if we're going to do RNA splicing, first we have to have phosphorylation of the RNA polymerase II tail, okay? Which is called the CDT, or the C-terminus domain, the CDT, okay, of the RNA polymerase II. And if you remember, we were just talking about it, the RNA polymerase II tail gets phosphorylated by transcription factor 2H. Okay, that's the one that's a kinase. You remember what a kinase is? That's right, it phosphorylates things, right? And when TF2H phosphorylates the RNA polymerase II tail, that triggers the splicing machinery to associate with the RNA polymerase. That's why the elongation is tied directly to the RNA splicing. Okay, but before we move on, let's back up and take a look at how the cap gets added. Okay, this is the first modification to the pre-mRNA. Okay, now, after our little baby mRNA transcript starts to grow out of the tunnel of the back of the RNA polymerase II, right? It's about 25 nucleotides long. It's just sort of peeking its head out of the RNA polymerase, right? And the 5' end of the transcript is popping out first, okay? And that 5' end gets a cap put on the end of it. And that cap is made of a modified guanine nucleotide, okay? So here's how it works. Basically, there are 3 simple steps to adding a cap. The first is that a phosphatase is going to remove a phosphate from the 5' end of the transcript, right? So that little phosphate on the end, we pull that phosphate off by a phosphatase, right? Kinases, they phosphorylate things, they put phosphates on. Phosphatases, they take a phosphate off, right? Okay, the second thing that happens in capping is a guanyl transferase. That's right, a guanyl transferase is going to add a GMP, sort of like in a reverse linkage, right? Back that truck up, add that GMP, right? Not a GTP, not a GDP, but a GMP. And it's going to add that GMP to the end of the transcript, right? So it's a 5' to 5' linkage instead of a 5' to 3'. Okay, then the third thing that happens is a methyl transferase is going to add a methyl group, okay, to the guanosine that we just added, right? Or that GMP. And that methyl group then makes a 7-methyl-guanosine, right? 7-methyl-guanosine. Okay, so again, 3 simple steps to capping. 1, we use a phosphatase, right, to move the phosphate on the end. 2, we use a guanyl transferase, adds a GMP in a reverse linkage, 5' to 5'. And 3, the methyl transferase adds a methyl group to the GMP, or, making 7-methyl-guanosine. Okay, now all three of those enzymes, all three of them are attached to the tail of the RNA polymerase II. Okay, now RNA polymerase I and RNA polymerase III, they make